Welcome everyone to another episode of Interviews with Authors. Today we're going to be discussing the paper Multi-Species cryo-EM Calibration and Workflow Verification Standard. I'm John Aguirre from the University of York, and today connecting from Madrid we've got Mark Van Rey. Hello Mark.
Hello.
How are you doing?
Very well, yeah.
Also we've got Cristina Nata from Sao Paulo.
Hi, hello from Brazil.
Is it sunny?
Very warm here.
Completely different picture here in York. Right, and the authors of the paper, we're going to call them by name so they can introduce themselves. Deja?
Hi, I'm Deja Bobet. I work as a staff microscopist at New York Structural Biology Center, and I helped carry out experiments, write the paper, and edit it.
Fantastic, thank you. And Jessalyn?
Hello, I'm Jessalyn Miller. I am a staff scientist microscopist at the New York Structural Biology Center, and I helped write the paper.
Thank you, Jessalyn. To conclude, we've got Ed.
Hi, I'm Ed Ng. I'm a senior scientist at the New York Structural Biology Center, and I also manage the National Center for Cryo-EM Access and Training, and this paper was my idea.
Thank you very much. Right, so the first question is from Christy.
Yes, so my question goes to Eddie. Eddie, what is a calibration standard and what problems will your calibration standard solve for the cryo-EM community?
Well, starting in 2017, when the Nobel Prize was awarded to Jacques Dubrichet, Richard Henderson, Joachim Frank, we had a lot of new people entering the cryo-EM field, and there was an explosion of instrumentation being installed. And what we were lacking was workflow standards and validation standards. So you had a lot of new practitioners trying to make use of cryo-EM technologies, but there were no calibration standards. And this calibration standard was hopefully to address cryo-EM workflows and benchmarking instrumentation.
Great, thank you.
Next question is from me. How did you select the bunch of macromolecules that compose the calibration standard?
Yeah, so we really wanted just to add first samples that we typically use for benchmarking our microscopes. So we had apiferidin, which is highly used in the field, and then some VLPs, which we also use for pixel size calibration. And then we wanted to diversify the symmetry since it's octahedral, icosahedral, very high symmetry. So we wanted something a little lower. So we started off with thyroglobulin, but that ended up being not so great because in cryo without a detergent, their floppy end bits kind of just fall apart. So we switched out to beta-gal. And then we also wanted to add in like something helical because just to be able to do that kind of processing, and we had TMV on hand. So
thank you. Sounds very interesting. You already explained one challenge. Did you face any more challenges when combining these samples?
Yeah, so at first it was kind of getting the ratios to work out. And we started off just in negative stain, but then translating that to cryo, it didn't necessarily look exactly how it did with those specific ratios in negative stain. So then we ended up just completely switching to cryo and doing everything there. But yeah, I think that took a little while to get right.
Yeah, I think you described also that the ice-water interface, or the thickness of the ice layer might affect also how many samples are, or how many of each of the samples are everywhere.
Yeah, exactly, because some are larger than others. So in the thicker ice, you would get most likely more of them, more of each species. But then in the thinner ice, you would get the smaller molecules like apiferidin or beta-gal.
Thank you. Very, very interesting.
Yes, but for the community, what can be evaluated using this calibration standard?
I think the main thing when people want a calibration standard, they want to keep things simple. But what we're seeing in cryo-EM is people are working with a wider variety of molecules and targets. So one sample can't do it all. So by incorporating multiple species, we can characterize protein complexes of different sizes, shapes, and structural complexities. And what that can do is they can not only assess the performance of their system, but their workflow. So not just their instrumentation, but their processing and annotation.
The paper mentions the potential for these standards to cater to a diverse array of biological samples. Could you please share some examples?
Yeah, so we chose an array of sizes and symmetries that would reflect the diversity of proteins that you might encounter just in, cryo-EM. So the smallest protein in our sample is beta-galactosidase. That's not a super small protein, but it is a lot lower contrast than the other proteins in the sample. And it's big enough that you should be able to get good resolution from a data set that's even collected on a 200 kV or even a lower powered microscope. So we didn't want to push the size too small. It's also really easy to freeze, really easy to identify. So if you're getting good beta-gal resolution, then other proteins in that size regime should also look good on your setup as well. We also included a virus-like protein to test polyhedral viruses and TMB for helical assemblies, such as bacterial filaments and filamentous phage. These have a really high symmetry, so you don't need many like boxed particles, but each particle has so many protein subunits that now determining the correct symmetry is the challenge.
Yeah, so a question for Deja. Do you foresee any specific collaborations or partnership that could increase the adoption of this standard in the scientific community?
Yeah, so the data set that we have already collected has been used in structural determination workshops. So I think it would be helpful if more workshops were able to use this data set. But it's a pretty small data set with a few proteins, so there's a lot you can do with it since it doesn't take up that much storage. But yeah, it's just a mix. We included some substitutions that you can do if you don't have the proteins on hand, if you wanted to make your own data set. So I think there's a lot of flexibility with what you can do with this data set, but you could also tweak it and make it your own.
Do you think that microscope vendors might send grids along with their microscopes with the sample? I'm thinking of toy microscopes that you buy in shops that come with a few slides of plant cells or something that you could maybe they might be interested in sending. I don't know if they do. I'm not a microscopist. I don't know. Maybe they already send around.
They don't send around, but they do sell. So Thermo Fisher sells apiferidin, and there's another company that sells membrane proteins and other targets. So I know people have been using them as benchmark samples. Well, because the microscope manufacturer installs and it's okay, you have a certain info limit, but at the end of the day, like, okay, I have a biological sample. Can it be used for that? And that's why the big pressure for a real biological sample to be tested.
Yeah.
What would be the number one target for getting the highest resolution, the number one candidate to send to the users of a new microscope?
I think the number one sample currently in the field is apiferidin. It has high symmetry, so it's very efficient for data collection. Yeah, and I think people have gotten close to 1 angstrom with it.
Fantastic, thank you.
And how can this standard be used to empower new researchers in cryo-EM?
We mentioned this briefly, but the education aspect is very important because as people are getting used to what EM data looks like and getting used to the software, you want to know, can you deal with heterogeneous samples? Can you deal with flexibility? Can you deal with a wide variety of factors that you might not be considering? And another thing is, in terms of instrumentation, our facility has many of the same types of instruments. We have 7 Cryst instruments, and next year we'll have 9 Cryst instruments. And you want to make sure that each instrument is performing not only within our facility, within other facilities. And so this is a standard that people can have a standard operating procedure for. We started this a long time ago, and I think if it wasn't for Deja and Jesslyn, it would still just be an idea.
Yeah, especially from one of your emails, I understood that Jesselyn was very important in finishing off the writing of the paper, especially. That sounds very relatable. If she wants to come a few months to our lab to do the same thing, that would be...
Yeah, most of the data, I think the whole data set was collected before I even worked at New York Structural Biology Center. So I was just kind of looking for something, just looking for a project and like, hey, this paper, just, kind of give it a boost.
Funny enough, you were quoting the resolutions. Our raw data was already gone. So that's how old the data was. So we only had the pre-processed data to work off of.
Oh, okay. So a little bit about the place you work. So you are also a service center. You obviously do your own research, and you're also a service center. How many users you have, more or less?
Well, the New York Structural Biology Center has three modalities. We have a beamline out in Brookhaven, we have an NMR facility, and we're in the EM facility. So our EM facility is one of the largest EM facilities in North America. We have, well, starting next year, we'll have 9 Cryos instruments, 4 Glacios instruments, and 3 dual beams. Jesselyn and Deja are part of our operations team, and they help users day in, day out. We have registered about 2,000 users. I think really only about 1,200 are active because there are a lot of people who register and we don't know if they're active or not. And we have, several hundred projects per year, anywhere between 700 to 800 projects per year. And we do a bit of research. So we have some flex time for that.
Mm-hmm. And I guess users can have different modalities. If there are some projects you might do almost everything. Other projects, you might just help them to put the sample in the microscope and they do everything else themselves. And I guess.
Right. A big initiative we have right now is trying to reach the nation and try to reach people who don't have cryo-EM resources. So then that becomes very involved. Where Deja is coming from, Deja can speak for herself. She's more in the tomography field, and that's a newer modality of cryo-EM. So maybe you want to speak about that.
Yeah, I was just wondering if you have international users as well.
We do. We actually had a user from Brazil a couple of months back. I don't know. It took him a lot of time because he was trying to get a visa to get over to the US, and he was saying that there were some riots in São Paulo or whatnot, and he couldn't get his visa, and so it was quite delayed. So it took him about a year or so to get the visa to come over to the US.
Big country, but it's small world.
I have a question for our younger contributors. What's next for you after this paper?
So I work for the National Center for In Situ Tomographic Ultramicroscopy. It's a mouthful, but in short, it's in situ. Very clever. And we basically are just a national cryo-ET centers. We're one spoke of 3, and then there's a main hub. So the hub is in Madison, Wisconsin, and then the spokes are at Slack in Colorado Boulder and us. And we basically work very similar to NCCAT, the National Center for basically single particle collection. So we're just their sister center. And we have, I think maybe around 40 projects now. It's been open for about 2 years. It goes through a proposal application process. It gets reviewed, and then they get sent out to the centers. So currently we just have an Aquilus 2 and high pressure freezers and a Leica EMGP to do sample preparation and milling. And then normally we send out the Lamella to Wisconsin, which has the Cryoses. So they do all of the data collection there.
Oh, fantastic. Thank you.
So do most users apply for funding?
We're more proposal based. So we have a regional center, and then we also have the national center network that Deja was mentioning. So the NIH Common Fund started 3 service centers around the nation, 1 in New York, 1 in Portland, and 1 in Stanford, and then created the TOMO network with the hub being in Madison and spokes again being in New York, Stanford, and Colorado. So these 7 service centers as a whole is helping the nation in cryo-EM, both in cryo-EM and cryo-ET. And then concurrently, they created 5 curriculum development centers for education. And we work very synergistically. So the national center is at no cost at the point of access, meaning NIH is sponsoring the researchers' access. So for that, it goes for external peer review and et cetera. So this is a little bit more of a different type of program because it's helping jumpstart research in areas that don't necessarily do structural biology. And also it helps centralize resources because it's very hard to fund creation of facilities in every state. It'll just take a very, very long time. So if you centralize in hubs, then we can have outside impact. So the funding structure is a little bit different. The individual research is funded by their own grants, but the instrumentation access, there is this National Center program here in the US.
Maybe Justin or Daja could answer. Do you get mostly users that physically come, or you're mostly remote users? With synchrotrons now, it's more common that you just send the crystals.
Mhm.
Yeah, here it's a mix. I think it depends on what their project is asking for. So a lot of it is service. So either they will give me sample, I'll make grids, mill, and then ship it out to Madison, or it's more like training where people either just come in for a day or two, or maybe even a full week, and they'll learn the whole entire workflow.
Hmm.
So we have kind of those two modes of access. Ideally, they get trained here, and if they have an instrument back at their home institution, then they're able to take everything that they learned here and apply it over there, and then disseminate that information to everybody else that's using that instrument.
Yeah, for our local users, it's also a mix. For local users, they can kind of come back over and over and over again because they live here in New York. And so some people want to learn everything they can about microscopy, how to operate the microscope, how to interpret their images. So they will come in for training, and then some people just want to send grids. But it's more fun if people come in and learn from us.
What was the thought process behind the selection of the viral-like particles? And what impact had the rest of the samples on the selection of the VLP you wanted to include in the sample?
Yeah, so I know we already had them on site because we're in collaboration with MGFin, who kindly provided all the VLP samples. So since we wanted another standard besides apiferidin to add in the mix, VLPs are very good for doing pixel size calibration. So after adding that, then it kind of determined what else to add, because if we added a low symmetry protein, then maybe we could add something else different. But since we already had two very high symmetry proteins, we needed something a little lower. So that kind of just, and it's pretty large. So it's kind of in the middle of TMV and apiferidin and beta gal. So it kind of gave like a middle ground, but it's very useful as well.
Sounds great. Thank you. Okay, so we've reached the end of our interview. I would like to thank the authors, Jessalyn, Deja, and Ed, for coming to this interview and discussing their paper with us. Thank you very much. Bye.
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 Bye.